it going, folks? I'm Matt, and this is Photosynthetic. Welcome to another episode of Meet the Industry. Been a minute since we've done one of these, but today I gotta say I'm pretty fired up. This is an episode which is really taking the two cornerstones of my channel, and that is organic gardening and technology, and it's smashing them together in this wonderful product called the Microbiometer. And today I've got Dr. Judy Fitzpatrick and Laura Decker from Microbiometer here today to talk. Ladies, welcome to the show. It's an absolute pleasure to have you here.
It's an honor to be here. Thank you for having us, Matt. We love to get the word out about going organic.
Oh, it's really the one thing that can truly save our planet. I mean, plant some trees, do some organic gardening, and stop using those salts. But that being said, we're not here to talk about salts. We're here to talk about testing and this wonderful device that you have. But before we get into that, I was hoping that you two could both maybe introduce yourself a little bit, talk about your background.
Great. So I come from medical diagnostics, and I had a medical diagnostics company, and I sold it to a Canadian pharmaceutical company and started teaching. And I got a $600,000 National Science Foundation grant to train laboratory workers.
Oh, nice.
And I was working in quality assurance. And I met one of my co-founders, James Sotillo, and he introduced me to the need for being able to measure microbes, because only microbes can actually enable effective organic gardening.
Yes.
And he said, we really need a simple, easy test. And I don't need to send it to a lab, because by the time that soil goes to a lab, the results are not reliable. They die. I mean, imagine putting you in a coffin and sending you for 3 days to a lab and seeing how well you perform.
Absolutely.
So anyhow, then I started working on it, and it was started with an NSF grant at the university that at the college I was at. Yeah. And it's gone through a number of iterations, and Brady Trexler joined us. And unfortunately, our co-founder, James Sotillo, who maintained all of New York City's public spaces organically, he died a couple of years ago at 50. So it's Brady and I that are here to carry on his work.
So Judy and Brady designed the test. Brady in particular brought in the ability for the cell phone to be really the medium that reads the test, as opposed to a microscope or a lab or a spectrophotometer, which is not something people want to carry around in their pockets.
Right.
So the current iteration we have had a couple constraints. What we wanted it to be was low cost, and we're still working on getting the cost down so that people can really use it a lot. It had to be totally safe, so you could send it around the world. You didn't have to worry about contributing to polluting the soil with our test. And we also wanted it to be sort of easy for anyone to do. So the app has instructions, we have videos, and right now we sell in 20 countries. So I think a lot of people who are in regenerative agriculture know that a lot of the world is a little bit ahead of us. So probably 60% of our sales are international, and we ship it all over the place, and it's a lot of fun. People test their soil for microbial biomass to find out how healthy it is, and also to assess the changes to practice that they're doing if they're working. So if you add oxygenated water as a new practice, can you see in a couple of weeks that the microbes go up? And they should.
Excellent. And yeah, this is the type of testing that I don't think anybody else is really doing, is providing a box for. And that's specifically what this box does. Is it measures the amount.
Box. We're not joking.
the amount of microbial activity in your soil, and if it's more bacterial or fungally dominant. Is that correct?
Yes. Mm-hmm.
Awesome. So what was the idea that really sparked this? Like, you kind of talked about it a little bit, Dr. Fitzpatrick, but how did you come to realize that, you needed this type of test device?
Well, the microbes are stuck to the soil very, very tightly. And that's wonderful, because the nutrients that are in the microbes then don't wash away. If you put down chemicals, when it rains, they're just going to wash away.
Yep.
But the microbes are stuck to the soil. And the wonderful thing about the glue that they make that sticks them to the soil is that that's what gives soil its texture. And when you have that texture, your soil will retain at least 50% more water, and it doesn't erode. And the earth has lost 50% of its soil. When we go in and test the soil in these farms where they're just using heavy NPK, the microbes are always below 100 micrograms microbial biomass per gram of soil, and the soil is just like sand.
Yeah.
It blows away. You know, it runs away when it rains. It's you know, and when they pour the chemicals on, the chemicals just go right straight through into the groundwater, so that you can't even use the groundwater in a lot of the states where there's heavy agriculture. And the Mississippi River is a mess.
Yeah.
And there's a dead area where the Mississippi enters into the Gulf, all because of this chemical runoff. Okay.
We have a little example of some of the solution getting separated. You want to separate the microbes from the soil in order to be able to isolate them to test them.
Right, right. And this is all about doing real-time testing, and that's important because you touched on that earlier. The microbes won't survive getting to a lab. So we talked about doing a demo. Maybe this is a good time to go through this and show us how the microbiometer actually works and measures this microbial activity.
Absolutely. And we have it here. And just like on a cooking show, we're going to fast forward through some of it and bring a cookie baked out of it. So we also have a demo on our YouTube page that goes a little bit faster. But what we'll say is the things that are important when you do the test and why they're important in test development. So the first thing is that you need to go out and get field moist, fresh soil. Okay. You're not taking a big core that goes 12 inches. Microbes live where plants grow and around the roots. So that's where you want to get the soil. Okay. You get the soil between the top 2 to 5 inches. And Judy has to chime in because she's the one who knows the technical side. And then we use these little sieves because, of course, if you have really good soil, it's going to have root hairs in it, decomposing matter. So you sift out the soil, right? And the video is there. And then you use this. The other thing that we did is we tried to take materials that were easy to get and manufactured so that we wouldn't have supply chain issues. Now, this is something that Judy predicted well before COVID, but it really meant that we didn't have any problems. So this is actually just a standard syringe that we cut the tip off, but it's a very effective way of measuring half a cc of soil.
Oh, very nice.
Okay, and they're quite easy to get. They're very low cost wise. You then take the soil, let's pretend it's in here. You put it in the test tube that we provide with a measurer. You only need clean water. It doesn't have to be distilled. It shouldn't be from a pond that has microbes in it, unless you want to test that pond. And then you put the extraction powder, which again is a sodium solution. So it is safe. You shouldn't eat it, but if you do, nothing bad will happen to you.
So just a quick question then. I mean, you talked about using just regular water. I'm assuming that if you want to use tap water, that's okay, as long as it doesn't have chloramine. And if it's just chlorine, you've off-gassed that for an appropriate
It
period
can
of
have
time.
chlorine. It can have
okay.
chlorine because the test is done in 20 minutes. Nothing's going to happen in 20 minutes.
Okay, interesting.
Okay.
Interesting. Yeah.
And, you know, if you have 1 or 2 microbes in your soil from the water you're using, they're, you know, as long as the water's clear, it's fine.
Good to know.
And then once you've got the soil in the tube, we actually, again, another design thing was that Judy, I know this doesn't look special. This is actually, people will recognize it. It's a frother that you use to make espresso. Again, they're readily available. They're fairly low expensive. They have just regular batteries because, of course, you can't mail around a crazy battery. And then we made a special probe, which I know it does not seem special to people, but it took her literally years to design because it's the perfect size to fit in the tube, to whisk to whisk enough to separate the microbes from the dirt, but not enough to sort of blow everything out of the water. So the one problem we have, home people do this test beautifully. Lab-based people sometimes say, I have a shaker, I have a test tube, and they use their own equipment and it just doesn't work. The simplicity is a little misleading because it is important that you do it, and it's not hard. Just use what's in the kit. So you whisk up the soil and then you let it settle, and you can see that the soil mineral particles go to the bottom, and this soil is a microbe-rich soil.
What I'd like to point out is, that we took a half cc of soil, which looks even smaller than the brown at the bottom, which is the soil, and look at how dark it is.
Yeah.
Every dark spot there is a microbe. That's how much microbes is in that tiny amount of soil. It's 1 billion microbes.
That's just amazing. So I guess the question, I grow my plants. I've got the big no-till 80-gallon beds. How is it that a small sample is representative of such a large container?
That is an interesting question, and we've been out there and I've done a bunch of fields like in Tennessee and we test all the time, and it's amazingly even, throughout the whole field. If it's dry, you have fewer microbes than if it's wet, but even so, we see like a 10% difference throughout an acre field. I mean, now if you have an area of the field that has dead plants in it, that's going to look different. But as long as things kind of look the same, then they pretty much in the same area read the same. On the other hand, right by us, somebody put in a mansion and had to put up trees to keep their neighbors from that. And they put a whole bunch of trees in. The trees cost $3,000 each. And in one spot, they have replaced the tree every year.
Really?
And we test the soil there, and the soil is 100. And on either side, where there's healthy trees, the soil is reading 400 or 500.
And that's
so there's
like
something in that there's something in that hole. I don't know what it is. Somebody said maybe there's an electrical wire that has is letting something go, or maybe somebody put some poison down there a long time ago or something. But it's interesting. I mean, if I were going to plant a $3,000 tree, I'd definitely test that soil first, and I wouldn't put it in until my soil looked pretty good.
When you're in a vineyard or someplace with trees, without sort of cover crops between, you will have a difference at the roots of the trees compared to the not necessarily. Okay. And pastures sometimes, because pastures are different, because when you put down organic matter, the microbes multiply. And because of what's happening in a pasture, you have cows pooping, there's more organic matter in some areas than there is in others. So you're going to see more variation within a pasture than you are in a well-maintained area. But the microbes are a colony, right? So there's many kinds of microbes, many kinds of fungi, and they do travel around. So that's why if you have a field that's fairly homogeneous in terms of rainwater and everything and what's happening to it, it should be fairly consistent.
Oh, that's good to know.
Yeah, so we took this and we let it settle. In the app, there are instructions and even timers. So you just hit timer, let it go off when the 20 minutes is up. And then what you wind up with is you take this has some stuff floating on the top because it's a garden soil, so there's a lot of mulch. You take this in and you get some solution in it, and you drop 3 drops carefully on this card.
Okay, just a little bit.
Yeah, 3 drops on the card.
So the card is interesting. It's a membrane inside plastic with an eyeball around the outside, an iris. And the iris is contained, it's called a grayscale, but a grayscale actually has a lot of different colors in it. So we can measure like how the light interacts with each one of these, and we compare that to the way the light is interacting with our microbes. And we can pick them up from their wavelength.
Wow, that's just fascinating. I know quite a bit about light and science behind that just because of my background in electronics. So to understand that this is all done using, like, it's just refraction of light off the microbes, and you're getting the wavelength coming back.
And we're doing a lot of different comparisons with the membrane, which is covered by the microbes, because the membrane has very small pores, so the microbes stay on the top of the membrane. And then we measure that and we put them on a card.
So this is another thing that doesn't seem like a big deal to people, but it is. When you're using a cell phone camera, we need to make the background the same. So you have a card that looks like this, that has a little picture of where you put it.
Yep.
And you put the card here and you use it. And that means that every camera and the flash has to be enabled, because again, that makes the setting the same, right?
Uniformity, yeah.
Uniformity. So the card, they're little coasters. We send tons of them out. They're super cheap, but that makes them the same. And then and I'll just show, people can go, here's a little, oh, you can't see my app, but here's my cute little app.
I'll put some stuff up on screen here.
Yeah, we have it. And then the app, you can see the light. It tells you when you took the test and you get the results right away. I will text this to you because you can't really see it.
Yeah, we'll put it up on screen and stuff here.
You put it up on screen.
Yeah.
And this is the beta test, but we put a new measure that sort of says, if you're doing agricultural soil, where do you fall sort of in the spectrum of things, and fungal to bacteria? And then you can hit a little button and text or email it to someone. So I'll do that to you and you can put this up. But that's it.
And it's just that simple.
And then this is totally safe solution. You don't have to worry about disposing it. You could put it down the sink or in the garbage or on the ground. There's nothing in it that's toxic. And that was, again, very important that we're not, some of the lab tests use pretty scary chemicals. And people aren't going to use chloroform at home, certainly.
The reason this is so new is it replaces a $550 lab test. And the $550 lab test, called chloroform fumigation extraction, takes a week and uses really toxic chemicals. Okay, and that test does not give you the fungal to bacterial ratio. And just to brag a little bit, okay, we are the only ones in the world who have actually enabled reading a card like this, so that you actually have a cell phone assay. Most of them have an instrument, and the instrument just transfers some data to your cell phone. This is actually run all over the world on all different kinds of cell phones, which is, what Brady did was amazing. Okay, under all kinds of light, sunlight, basement, wherever,
I was going to ask, about ambient lighting.
Since you know about photonics, you know how difficult that is. Yeah. So we will say, that a lot of the value we have is in the app that we have that actually allows you to store the data, so that you don't have to say, where is my result when May? Yeah. You can see that.
Yeah, the app will GPS locate your sample, date and time stamp it, and it has fields that you can put in little notes like a week after I put down compost tea, or any information you want. And then you can download them to Excel if you're doing a lot of data analysis, or you can just refer them in the future without having to remember or have a bunch of pieces of paper.
I love it. It makes so much sense. And I mean, you've touched so many brilliant points. I mean, it's easy, it's repeatable, it's environmentally friendly, it's not very expensive. This is fantastic. So let's talk about results, though. What do the numbers mean? When I'm looking at the screen, what are these numbers telling me?
Well, if you're not using chemicals, which people did for a long, long time, even gardeners, okay, then the nutrients you have to bring to your plant have to come from the microbes.
Yes.
Okay, the microbes actually barter with the plant. Okay, so if a plant wants more phosphorus from a fungus, it gives it more food. Okay, sometimes another plant will actually compete and offer even more food. And then the fungus sends more phosphorus to that other plant. Okay, so you have a whole community of plants here. They're going to bring all the, minerals and everything to it. And then the really, really important thing, I think, for organic, people who are interested in organic, is the immune system of the plant are the microbes.
Yes.
Okay, the microbes actually enter the plant through a process called rhizophagy. Rhizo means root, and phagy means eat. So I don't know if they actually eat them, but the microbes go into the plant, and once they're in the plant, they stimulate the plant to form root hairs. What's good about a root hair is it enlarges the amount of root that can absorb water and nutrients from the soil. Okay, and while those microbes are in there, they contribute 40% of the nitrogen that the plant needs. And the microbial population is capable of fixing nitrogen from the air. And it can mine, mineral nutrients from the soil that don't show up on chemical tests. So some of our organic farmers will say, they tell me my soil's, deficient in this and this and this, but my produce isn't.
Yeah.
So the only way it can get into your produce is through your microbes. So I have to, and we know right now that there's way too much phosphorus in the soils of the earth. They just keep putting down phosphorus. And when you put the chemical down, it washes away, or it becomes insoluble, okay, to the plant, unless it's delivered to them by the fungi.
Yep.
And the fungi they depend on the bacteria and the amoeba and the other things in the system. This is a very interdependent system. I mean, I'm just sitting here saying to myself, imagine a baker who has no access to flour, no access to yeast, no salt, nobody to bring, no customers. We have to take in certain nutrients, right? There's vitamins and things that we can't make for ourselves, even if we eat it. Can imagine the microbe has like 1/20th or 1/50th of the genes we have. They're much more dependent on one another for the nutrients just the microbes themselves need to form that community. So, you're building a community. And the other interesting thing I've been thinking about is, when the microbes are, giving the soil texture, what they're actually doing is making a home for themselves.
Yep.
Okay, because then you get, like little micro spaces, little caves and the microbes like to live in there because it protects them. Like amoeba eat a lot of bacteria every single day, but the bacteria that can hide inside these little places, okay, are saved from them. So, you have a whole system that's like very, very dependent on one another. They show you the pyramid all the time, and the bottom are the microbes. What they don't tell you is the next stage, it has only 1/1000th the amount of biological material in it. So, if there's like one ant here, 1,000 ants here, there's only one here. And it keeps going like that at each stage of the food web, because the whole food web in the soil, worms and everything else, depend on having a large microbial population there.
Absolutely.
And if you're an organic farmer, they did a big study in Europe, and what they showed from about 500 farms that they looked at over many years, this was published in Nature, 97% less pesticides.
Yeah.
Okay. And one of the interesting things about that is this plant meets pathogens, the organically treated. It's like a kid who goes to nursery school. Okay, they get a little cold. That kid who goes to nursery school and gets a little cold develops a better immune system than the kid who isn't exposed to any disease. Okay, and that immune system is over 2,000 antioxidants. Okay, and those antioxidants are what gives your food its flavor, because everybody knows organic food has more flavor. In every test, they show that. And the nutrients that you get from there that are anti-carcinogenic, et cetera, okay? None of those is measured by the USDA. The USDA says organic food and non-organic food have the same level of nutrients. They're only looking at like carbohydrates, fats, and proteins.
Right.
They're not looking at all these many compounds that are so very healthful and are grown because the plant is working with the microbes, okay? And together they're keeping, diseases of the soil and of the plant down.
Yeah. And we will say that the antioxidants and the immunity is also what gives medicinal plants a lot of its variety, its taste, and its smell and its qualities. So that's why a lot of medicinal plant growers are very, very into getting their fungal content high, because those are the ones that go up it and give the plant really the nuances that you're looking for in it. I'll also say, and people ask all the time, they get a result on the microbiometer and say the number is 300, right? And a lot of them say, what do we do? And part of the problem with traditional agriculture, right, is that chemical companies gave you this thing and they said, oh, now you throw down nitrogen or phosphorus. And it's more of an art. And we're sorry, we wish we could tell everyone on earth what to do to heal their soil, but it's a very hard question. So what we like to tell people to do is to use the microbiometer as a benchmark test, right? Today, and it depends on the life cycle of the soil. You do know that in the middle of winter and early spring, your microbes are going to be different. But if you test, and if you're indoors, there's less variety. What you want to do is test your soil at the same point in the growing cycle and say, I'm testing it, and let me see what changes there are. And we do know that you want a higher microbial content because it leads to more diversity and more strength and more resiliency. So in general, agricultural soil, anything below 200 is quite poor. But for example, in some place like Australia, 200 might be kind of all the land can sustain. And 600 in agricultural soil is quite good. So I live in the Pacific Northwest. If I go out to my gardens that I, mulch, they'll be 600 to 700, right? But if you're growing medicinal plants inside and you're using compost tea and all kinds of you leave the roots in, you don't ever sterilize it, we've seen numbers around 2,500. So, I mean, those are just engines of soil. So it really depends, but it shouldn't be considered good, bad, right? That's not necessarily helpful. What you want to say is, am I moving along the right way? Am I protecting my soil? And am I not just feeding my plant, but am I feeding my plant microbes so that they can feed my?
Right. So you take the app, you do your first test. You're basically setting a benchmark of a number. It's not really a number based off units. It's just more of a count, and then you're looking for over time that count to increase, and that's going to show you the health of your soil. But I guess the question is, how do I tell if it's more fungal or bacterial dominant, and why does that matter?
Well, the test will return to you a fungal to bacteria ratio, right?
Oh, okay.
Yeah, as soon as you get it, it'll come back. And Judy might need to correct me, but I think in general, if you're agricultural outside soil, a 1-to-1 is pretty common, right? If you're growing medicinal plants indoors, or perhaps you're growing strawberries or other very berries, you want a higher fungal content. For example, vineyards with grapes want a higher fungal content because it contributes so much to the quality of their plant. So you get it and say you benchmark, you say, okay, here my container is now 60% fungal, 40% bacterial. I'm going to put down some mycorrhizal fungi. I'm going to wait a week or so. I'm going to test it again. I did that experiment at home. It was really fun. And when I did colonize one plant with mycorrhizal fungi, both, and waited 5 days, I tested them both again. The fungal to bacteria, the fungal ratio went up a lot in the one I colonized, but so did the total microbial count.
Oh, interesting.
Yeah, which meant, obviously the fungi grew up a lot, so it did both.
Right.
The same thing if you're using another amendment, you can use the test to you use a slightly different technique, but it's in the app, to do compost tea. To say
that was going to be my question can I do this to measure compost tea activity?
Now, the funny thing about the scale of compost tea, because we test it a little differently, you actually just put the tea in here with the solution, is a number like 20 for compost tea is quite good.
Oh, okay.
It depends on the dilution of the compost tea. I mean, you can make it very strong, and then people usually dilute it. So it's going to depend on how much you've diluted it, what your reading's going to be.
Oh, that's fascinating. Very, very fascinating.
But if you're using the same compost tea over time, or you're making your own, it's a really good way to say, I always get 20. This batch I got 6, something went wrong, or this batch I got 60. I came up with a new recipe.
Yeah. The ability to benchmark and then test again and test again. That's awesome. Hey, so the frequency of testing, what would be considered average or maybe too much?
It's going to depend on what you're looking for.
Okay.
Like if you wanted to see, you can put down some AMF, okay, or some things, or some other biologicals that can actually harm your plant.
Of course.
So, you look a week later and things have gone down. This is not where you want to be. You can, in pots, people take and to check which cover crop is actually going to complement their soil the best. It's just like you go to the doctor, it doesn't have one pill for everything. Each soil has different problems and different things that it can use. So when you try all these different cover crops or cover crop mixes and do it in a pot, within a month you can see, which, just by looking at the microbial count, which is the one that's supplying what the soil is missing.
Right.
Because if the soil is missing nitrogen, then if you put down clover, because it brings in a lot of nitrogen, and you're going to see like the carbon sponge in New York saw a 600% increase in the microbes in 3 weeks using
That's
clover
insane.
On their soil.
Yeah. That makes sense.
We're not
though.
saying, but then people say, oh, I should always put down clover, right?
Nope.
Well, that's not true.
Yeah, in that
have to
particular circumstance, that's what works for that.
And,
yeah,
you have to do the hard work. So the point of the test is we're giving you a tool to help guide you as you experiment with your soil, because there's not a roadmap that's out there, and what agronomists have been giving us is not a good idea. We need to stop doing that.
Yeah. Well, and I talk about this a lot too, that what I do to grow my plants works well for me because my environment, my situation, my inputs, the way I do everything, and that's not going to be the same for somebody else. So to take that ability and be able to go, I did a thing, my plants look like this, I can take and look at the activity in the soil and equate that all to either good or bad results is an excellent way to proceed, especially for newer growers too. I would think this would be a super valuable kit in their toolbox. Although I guess to that point, when we talk about affordability, and the cost of the device is I think it's about 130, 140 US.
Yeah, it's 100 to get the box, which is everything you need to sort of run the test except some tap water, is $135 free shipping, and then you don't need a box every time, of course, and you certainly don't want to keep filling the world with these plastic boxes. We get refills, and the refills will go down. You can get them down as low as $7 each to run them.
Yeah, that's fantastic. That's
and
fantastic.
as we build sales, we actually would like to drive the price lower because it is an experimental tool, and we want people to be able to use it, in exciting ways. So yeah, it's about $7 a test, and it takes 20 minutes. So you don't have to go ship it off somewhere or do any of that.
Well, instant and being able to repeat it when you need it. But I guess if somebody is just starting out, and this might be, one of those things that they can't achieve, is there a way to test or look at things that they can do to determine if their soil is more fungal or bacterial dominated? And could you speak to that a little bit?
I mean, another method of testing your soil for fungal to bacteria?
Yeah, for the home grower who's just getting started.
Okay, without using our test.
Right.
Okay, microscope.
Now, the microscope is very, very difficult. There's basically PLFA is, most people don't really believe in it anymore as a way of seeing fungal. And bacterial, when they do it, the fungi, whenever they get, they're very fragile. So they break up.
Okay.
And they're different sizes. So you're getting like small tubes, or you're getting spores. And we pick them up, because they absorb at different wavelengths than the bacteria. And we have patent on that. But otherwise, people actually count them by counting many, many, many slides and measuring the length of it and the width of it, and assuming that you're only seeing about 1/100th or less of what's there. So you multiply by a big number.
Yeah, that makes sense.
So that gives, so if a person has spent their life doing this, they're usually pretty good at it. There's a couple of people in the world who are very good at it.
Yeah.
But it's almost impossible. What we do is we've validated by microscopy, and we can look at size because the microbes are under 2 microns, and the fungi are generally bigger.
Yeah, and microscopes are important, but I mean, if folks can look at that little center, it's not particularly big. But when you compare that to what's in the field of your vision when you're looking at a slide, it's hundreds of times bigger.
Yeah.
So it does give us a much, even though it's not quite, it doesn't look very big, it's much bigger than you can get from microscopy. And there are other companies that do more genetic testing, DNA analysis to do fungal to bacteria. Again, a lot of those involve shipping it, right? And so it's unclear what happens in that process. And then cost is certainly an issue. But we all know that the ultimate answer is how good is your plant, right?
That's the thing too,
But you don't want to wait till you've killed it because you did the wrong thing. So
yeah, absolutely. Well, I mean, just a couple of things you touched on there too, like microscope testing, more lab testing, and these are all things that are going to cost additional money and whatnot. So when you look at the cost of a microscope and learning how to do all of this testing versus just the kit that you offer, I can see it being definitely the more affordable solution. Now, what if I'm growing in something like coco coir or pit moss? Can I do the testing in there? Organic growing, just not soil.
Yeah, we do that. And I use coco coir and peat moss. Okay. And actually both of those have a lot of microbes in them to begin with.
Right.
So you can actually test coco coir, or you can test peat moss and see how much is in it. What we can't tell you is, we can't tell you exactly what's wrong with your soil. Okay.
I think that makes sense too. It's just giving that count.
Yeah, people, Laura's brought up hydroponics.
Right.
in hydroponics, generally speaking, the microbes have no color. The pigments that we see in microbes in the soil, in the top 3 to 5 inches of the soil, are not present in some composts that people make under strange conditions, you know, that are under like laboratory conditions. We can't pick them up because they're not colored. And the color of the microbes does not correlate with like the color of the soil. So we're not influenced by the color of the soil, because some soils are yellows, some are red. Now, if people have a heavy red clay soil, which is unusual, we include in our kit a magnet.
Oh, okay. Pull the iron out.
And under those conditions, it would be pink.
Right.
But if you let it settle for the 20 minutes on the magnet, those are like tiny, tiny iron particles. They will precipitate, and we can actually look at how many microbes are there. So, well, clay is like really good for, some clay in soil is excellent because it provides a lot of minerals that the microbes and the plant need. Heavy clay is usually low because it doesn't have much oxygen. It's just too dense, and you can't sift heavy clay.
No.
We tried.
You could go crazy trying to,
Yeah. The other thing is the test is actually designed for microbes in the earth. So some of the very, very complex scientific, hydroponics that are all clear or using unusual things are, we haven't done enough testing for it. The same is true as if you make a compost with very weird things in it. You need to be a little bit careful that you're not, either putting too much or too little pigmentation in.
Well, we've checked a lot of the things that people put in composts, like humic acid and molasses. And at the levels that people put those in compost cheese, we don't have any interference in the assay.
Yeah, it's negligible at best then.
Yeah,
so makes a lot of sense. Now, what about?
And now we have never seen a sick plant with healthy microbial count.
Makes sense. Makes absolute sense because the soil is doing everything that the plant needs for it.
Well, I mean, in a way, it's kind of confusing because, usually if you think of a sick person, you think they're going to have a microbial infection. You think it might be high.
Yeah.
But it's not.
No, the bad bacteria for sure.
The bad ones, they seem to kill off the good ones.
Yeah, this is true. Now, for the kit itself, is there any real maintenance required when you're done doing your testing? Do you have to clean slides? Can you speak to that a little bit?
Yeah, I mean, we have a green refill, which we encourage people to use, and you take the tube and you rinse it out. It's fine. You don't have to disinfect it or do anything crazy. You do occasionally, when you get a refill, you get a new probe. These are a little delicate. They sometimes can break. So these probes are provided. And then sometimes you have to change the battery in your coffee whisker. But that's about it. We do keep the test cards are a little fussy, so they come with a little case.
Okay.
And when you get the refills, they come in a bag and you put them in the case just to keep them from getting scratched and things like that. But there's again no crazy parts that you need. And the refills you can just throw right in the box that you had, because the refills will primarily be the extraction powder and the cards.
Right. And there's nothing that needs calibration either that I can tell. And that's the wonderful thing, because background in testing and whatnot, always suspect of your equipment, especially if it has a calibration sticker on it. So, I mean, it ticks all the right boxes. It's affordable, sustainable. I mean, everything. I just love it. So I guess as we get to the end of the interview here, I will put links down below where people can find it. You guys did give me a discount code as well, guys. So head on over there and you can pick up your own. But I guess last question today, ladies. What's on the horizon for the Microbiometer? Is there anything new coming out or events that you're going to be at that we can find you?
Yeah, we will be in December of 2021 at the Acres Convention in Cincinnati. I am looking to go to the Jacob Javits Center, I think in January for their show. I forgot the name of it. And then the things that we're doing are really upgrades to the app. So we've added recently a function. How it used to be is you took a test, it was in your data file. But a lot of people do it with other people. So we put in a project management function.
Oh, nice.
So you can create a project, invite all kinds of different people to put samples into the database so everyone can see it instead of having to manually put it together. And then we're also adding a function. A lot of people go to the field, pick up the soil, take it back to their living room and test it. And we're setting a function so that while you're in the field, you can make the sample so that the GPS is identical to where you took the soil. So for repeatability, right, you can go to the spot again. And we'd like a little map function. So we're trying to make, again, people are using it, they're getting a lot of data, and we want to make sure that that data is available to them and correct in all the ways they need it, so that year over year they can look at it.
I think one of the things that we're working with a number of academic institutions, universities, and they're looking at, like, why is there's 2 million articles saying microbial biomass done by that $550 test is the best indicator of soil quality. Okay, and if it's a long-term managed property, it also tells you that you've increased your carbon, because the carbon that's stored in your soil is the body of dead microbes.
Yep.
So it's absolutely impossible to increase it without increasing your number of microbes. Okay, so what we're working on with people at universities is, what tests do we not need to do in a lab anymore? You know, what tests are replaceable by microbial biomass, so that we know the exact functions that we could begin to tell you that microbial. Now, we do know that that microbial biomass, if it's good, it tells you have adequate nitrogen. It tells you have adequate phosphorus. I mean, the correlations for those are like 90%. So yep. That's really good. Potassium is like 80%. So it is telling you, you know, if you don't have critical components, any critical component, your microbial biomass is going to be low.
Love it. Just
like if anything critical for a human is missing, you can't make it up by giving them pasta.
That's a great analogy.
And the other thing is we're a very small company. There's 4 of us. We all do everything. And on the app is our email. And so as people use it and find either problems or difficulties or things they want, people write to us all the time. And that's how we get most of our ideas. So it is very dependent upon people. Carbon is for agricultural growers going to be the next big crop. For farming to be sustainable, growers need to grow corn, wheat, and carbon. The carbon tests now are $500. People in Africa who have 5 acres cannot do that test, but they do need access to the carbon markets. So we're trying to get into studies to say this is a very, very good proxy audit tool for carbon sequestration that could be done. I mean, you need a cell phone, which everyone has in the world primarily, but you don't need to be on a network to take the test.
Yeah.
So you could be in the mountains and still take the test. And we're hoping that in terms of developing countries will really help the agricultural sector cost effectively be able to tap into carbon markets. But we need studies, and we need people to call us up and say they want to do it for us.
More science. Ladies, this has been an absolute eye-opening chat today. I certainly appreciate you taking the time to speak to me. And, to people watching, guys, go ahead, head over to their website, take a look at the device, and maybe it's going to be right for your garden. That being said, again, absolutely appreciate your time. Laura, Dr. Fitzpatrick, thank you so much for stopping by today.
Thank you. Enjoy.
Thank you for spreading the word.
Awesome. All right, we're out.
Okay.